I guess, one example would be genome architecture in different lineages. So bacteria have very, very streamlined genomes. They're almost 100% coding DNA, 95% plus, very little excess DNA between genes and so on. And then you move into eukaryotes and, a lot of excess DNA in between genes. Our genes are split up by things called introns. So our coding DNA are actually islands in a sea of intronic DNA that actually gets transcribed, but then has to be spliced out for to make a productive messenger RNA by a machine called the spliceosome. Bacteria don't have these kinds of introns. They don't have this kind of problem. Our genomes, especially in animals and land plants, are just littered with what are called mobile genetic elements, or some people call them jumping genes, transposable elements. The human genome is only about 1.5% coding DNA. So how did we get here? And why is it such a mess in us multicellular lineages and in eukaryotes in general of the bacteria? The idea is it was a consequence of a reduction in effective population sizes when the eukaryotic domain started to emerge, and then further reductions in multicellular lineages. That made it easier for these genomic changes to accumulate in an effectively neutral fashion.
Hmm. So,
So,
so if I'm hearing you correctly, basically, we when we compare simple organisms like prokaryotes to complicated organisms like eukaryotes, one of the things that what we mean by complexity in one way is genomic complexity. So um, we've got more stuff. There are more features to our genes. It's not just purely chunk of DNA coding protein. There's a bunch of stuff in the middle, like the introns and those non-coding elements that you mentioned. So I guess in principle, there's two ways you could start to think about that. One would be, okay, all this complexity must be doing something. It must be buying us something. It's been selected in because there's an advantage to having all that extra stuff. The other way to think about it would be that it's a because the populations are smaller in these types of organisms, this stuff just accumulates passively because selection can't really fine tune things and get rid of it. And it sounds like you're saying that a lot of it can be explained in that way, that it's just the natural passive accumulation of stuff based on population size.
yeah, that's basically my view of all this. It's a passive expansion of excess DNA in genomes. Now, if the mutation bias was for deletions, things would go the other way and in sufficiently small populations. You're just being pushed by mutation pressure.
Mm.
And for mobile genetic elements, they have a built-in mutation pressure. They're copies of DNA, selfish DNA, that can make offspring of themselves and insert in large chunks in other regions of the genome. So that's why we're biased towards large size. But there is an interesting point to be made here is that we should make a distinction between the origins of alterations in something like genome architecture or any other feature of an organism, which could be by completely passive mechanisms, not advanced by adaptive mechanisms. But now you've sort of changed the genomic real estate, and you have, different kinds of material upon which natural selection might operate. It wasn't there before. A good example of this would be introns. As I said, our eukaryotic genes are in pieces, and in us multicellular species, the coding DNA are little tiny islands separated by these really large introns. And in multicellular lineages, these have become useful because it's possible to differentially splice out introns. So you can have alternatives, tissue-specific splicing of genes, and you can make different variants of genes that might work in one way in one tissue and be utilized in another way in a different tissue.
Mm-hmm.
So that's a way of thinking about how introns might be advantageous in certain lineages, in multicellular organisms with different tissues, for example. But introns evolved at the root of eukaryotes before there were multicellular organisms. So you can't invoke, tissue-specific alternative splicing as the
As
mechanism that, promoted intron
yeah,
evolution by natural selection. That came later.
I see. So it sounds like basically, um, due to the interaction between drift and selection and the population size, you can get these increases in genome size and complexity by completely passive means. It just sort of naturally accumulates over time. But in doing that, you're sort of creating a richer, more diverse substrate upon which selection can subsequently act. So you can take advantage of some of this complexity that emerges passively and now do things that you couldn't have done before it emerged.
Yeah. But you're going down a certain pathway that was, developed by non-adaptive mechanisms.
Mm-hmm.
That's a lot of what biology is all about is, historical events that, set us down a certain course forever, such as ATP synthase, a bizarre way for us to produce energy, but that was the state of affairs in the last universal common ancestor, and there's no way going back anymore. Same thing with the ribosome.
Hmm. And what does this say about the reproducibility or predictability of the evolutionary process? So, for example, if you go back to the origin of life, or you go back to the origin of eukaryotes, or you go back to the origin of animals, and then you let things play forward in time again for 500 million years or whatever, would you see similar things play out, or could it be completely different from that starting point?
Well, I don't know. I'm capable of that kind of crystal gazing, but um, I mean, there's a lot of other events to worry about here because it's the Earth's geological history too that has played a role here. So it's that's a very difficult question to answer, but drift is a chance process.
Mm-hmm.
And there have been historical contingencies where, for whatever reason, certain kinds of changes arose in an ancestor to very, very large groups of organisms for some vital function that can never be lost. So, in that sense, there can be these ratchet like effects. Many of those, I think, would probably be unpredictable in certain geological contexts, not knowing the particular settings.
Mm-hmm. Um, okay, but it sounds like there's a quite a large role for pure chance here.
Yeah, well, that's what I would certainly say. Sure.